Well, hello, ladies and gents, and welcome to this last installment looking at the macromolecules. So, I know as a teacher, I probably shouldn't have favorite macromolecules, but I can't help myself. Proteins are absolutely fantastic, just in the diversity of structures and all the cool things they're able to do. So, what I'd like to do is walk you through some of the amazing features of proteins. Now, in terms of features, you don't need to memorize it, it's more than 50% of the dry mass of cells. Just know that there are all sorts of different functions for proteins uh, within cells. So let's look at first one of the big features of proteins. It's that many of them act as enzymes. Now we know that enzymes are catalysts, that they speed up a reaction, but a fair question is by like how much, right? So what sort of impact do enzymes have on the rate of chemical reactions? And under optimal conditions, it's not always the case, but under optimal conditions, an enzyme can catalyze 400 or 500,000 reactions per second. So every second, an enzyme can catalyze hundreds of thousands of reactions. So without the capacity for enzymes to do the work they do, reactions in the body would not happen fast enough to sustain life. So enzymes are incredibly important because they can be reused again and again and again and again to speed up the rate of chemical reactions. Now, let's talk briefly about sort of some nuance in the structure of enzymes. I'm guessing you've heard of the lock and key model. The idea is an enzyme has an active site, the portion of the enzyme that actually is involved in the chemical reaction, that is highly particular. And a lot of times it's referred to as a lock and a key, like only a certain shape lock or key fits into a certain lock. And that was for a long time sort of the notion of how enzymes work. Today we have, again, a little bit more nuance in the view for how enzymes do their work. And I think the illustration does a good job of explaining that. You see the two substrates, the yellow and blue materials that attach to the active site of the enzyme. The active site is the one that's sort of scalloped and it's got the red stars by it. So notice that when the substrates dock on the active site, they interact with one another and form a product, right? But look more closely at the enzyme. Do you see how the active shape of the enzyme isn't a perfect fit for the substrates, but that when the substrates are aligned with the active site actually changes its shape slightly to better accommodate the substrates. That's the idea of what's called an induced fit. The idea of an induced fit, I think your book aptly describes it as being like a handshake. People's hands may approximate one another as they approach, but then they'll sort of grasp around one another and change their shape slightly to have a tighter fit. And that's exactly what happens with the active sites of enzymes. Once the product is produced and released, notice that the active site of the enzyme goes back to its original conformation or shape. So I think the idea of induced fit is good because it speaks to the specificity of enzymes, that enzymes only catalyze certain types of substrates. But induced fit gives maybe a truer sense of what happens to enzymes. They sort of reversibly change their shape to better accommodate or better fit the substrates that dock in their active sites. Okay, a couple things to look at with this. One is how naming is done. If you see something in an ose like sucrose is table sugar. It's a disaccharide made of fructose and glucose. But if it ends in ose, typically that's telling you it's a sugar. Sucrase is an enzyme. If it ends in ase, again, usually it's an enzyme. And enzymes are usually named, or not usually, oftentimes named after the substrates in which they act. So sucrose is the sugar. Sucrase is the enzyme that acts on sucrose and breaks it into its constituent monomers. All right, now we know that amino acids are the building blocks of proteins. Amino acids, I think of them as sort of like individual Lego pieces. So you have a distinct Lego piece, and there are 20 different pieces, and you can put them together in all sorts of different combinations and make all sorts of different Lego structures, right? Well, we've got 20 different amino acids, and you can link different combinations of amino acids together different ways, and you're going to create what are called polypeptides. A peptide is a name for the specific bond between two amino acids. An amino group combines or bonds with a carboxyl group of an adjacent amino acid, and it forms this peptide bond. So a polypeptide is literally just a chain of amino acids. And a protein is when the polypeptide folds up into a three-dimensional shape and is functional. So a polypeptide is the chain of amino acids, and we call it a protein once it's folded up and ready to get to work. So if you look at the sequence on the screen there, on the left, you see the chain of amino acids, right? And then it will end up folding into a three-dimensional shape and become an active protein. Now, look closely at the amino acid. It'll be helpful on the test to be able to identify the structure of amino acids. Amino acids, luckily, have a lot in common. Most amino acids all have many of the same basic features. They just differ in very particular ways. So, if you look at a particular amino acid, what I recommend is trying to first find what's called the alpha carbon. The alpha carbon is the carbon in the middle of the molecule that has 4 different partners. If we look at this alpha carbon here, it's got an amino group that it's covalently bonded to. An amino group has the nitrogen and a couple hydrogens. The alpha carbon is also bonded to a carboxyl group that has a single bond with the carbon and a double bond with the carbon. And then it'll have a hydrogen that they don't show there. The alpha carbon has a little hydrogen buddy off to the side. And then the alpha carbon has what's called an R group, or sometimes called a side chain. All the skeletal elements of amino acids are the same. Amino group, carboxyl group, hydrogen, alpha carbon. How amino acids differ is in their R group. The R group is like the flare that makes different amino acids stand out. And since R groups have different combinations of atoms, they'll have different chemical properties, and they'll make the amino acids fold up into a polypeptide in different ways. So here's another image that does a nice job of outlining the features of an amino acid. You have the alpha carbon in the middle. There's the amino group on one side, the carboxyl group on the other, the hydrogen buddy, and then there's the R group. So I like to show the combination of the juxtaposition of these two slides because sometimes the R group may be represented either above or below, just depending on how they choose to represent that. Okay, I'd like to pause here and introduce a good friend of mine. This is Dr. Chris Johnson. He and I have been friends since high school. He's actually a friend that introduced my wife and I to one another, so I owe him a huge debt of gratitude for the rest of my life. But I want to mention Chris because Chris is a molecular biologist, and he and I, talk biology all the time. And one thing that was interesting was when Chris was doing his PhD dissertation a number of years ago, he was talking about the experience he had in AP Bio, where Mrs. Dejan, the teacher at our school for AP Bio, she had described to students how proteins do the work in cells and how proteins have very specific shapes to allow them to do their jobs, and that DNA provides the instructions for how to assemble those proteins. So if you can manipulate the DNA, you can change how the proteins fold up, and you can make them do different jobs, or you can fix damaged proteins, and you can sort of change expression of traits. And Chris said that when he heard that, like his jaw dropped, and he was looking around the room because he just couldn't believe it. And he said a lot of the people just had sort of blank looks during the lecture, but that he said when he figured that out, when he learned that, he knew that that's what he wanted to spend the rest of his professional life working on. Like, he's like, this is what I want to do. And so Chris now works at NREL, that's the National Renewable Energy Lab in Golden, Colorado, doing all sorts of research on sort of environmental topics. So it's a really cool job. And it was fun because it was, I guess about a year and a half ago, Chris sent me a magazine article from Popular Science. His lab at NREL had been involved in some pretty cool research on an enzyme called PETase. Now, plastics are definitely an issue in the world, right? Plastics don't degrade readily, and when we recycle them, it's really sort of like a downcycling, right? You sort of cut up the plastic into smaller pieces and use it for something else that's a little flimsier, but there's really no good way to get rid of plastics. Well, a handful of years ago in Japan at a recycling center, researchers had identified an enzyme called PETase that cuts up the plastic polymer. There are bacteria in this recycling center that had evolved to be able to cut up plastics. So they could do it. The enzyme, this PETase enzyme, can cut up the polymers in plastics. It does it really, really slowly. So labs around the world are trying to figure out where did PETase come from. And so certain researchers noticed that the active site of PETase looks similar to another enzyme called cutinase. Cutinase cuts up the wax on leaves. So bacteria can produce this cutinase, remove the waxy protection on leaves, and then begin to digest the leaves, like the image that you see on the left there. So labs have been involved with trying to see if they could sort of reverse engineer PETase to be able to demonstrate that PETase evolved from this cutinase enzyme. And what his lab had done is accidentally, when they were trying to sort of reconfigure PETase, they created a version of PETase that was much more efficient, that could break down the plastics much more rapidly. So now NREL and others have this interest in trying to develop PETase to be able to digest plastics much more readily at a commercial scale. So hopefully, in the future, we'll be able to actually recycle the molecules in plastics and turn them into something new, rather than just being able to cut them up and sort of downcycle them. You can truly recycle them into brand new molecules. So this is like an absolutely breakthrough discovery. And now they just got to work on the metabolism to be able to crank it up to something that's, economically feasible. So I think it's an awesome example of how you can take someone who's, decent student in high school, but has serious interests and dedication to, learning about how the world works. And then, down the line, he's able to help play a role in a discovery that could potentially have huge ramifications for, the health of the planet. So I know that a lot of people enjoy like movies and stories about superheroes, but really it's the people in labs that are doing the work to figure out how nature works to make things better for all of us that are absolute heroes. So, if you are someone that thinks you want to get into the scientific realm, you can really have an impact on people in significant ways through the work that you do. So I'm 100% for, people going and pursuing their passions. I'll put a link to the scholarly article, like the journal article that they had printed, and then the popular science article as well. So if you guys want to read that, that's pretty interesting stuff. All right. So we know that, again, polypeptides are chains of amino acids. They'll fold up into a certain three-dimensional shape, and then we call it a protein. Now, if you change the order of the amino acids, well, then you can change the shape of the protein and change, whether or not it works or how well it works. So structure and function are really key in terms of understanding protein capacities. Here they show, these are sort of collapsed models of an antibody and the protein that it's interacting with. You can see how the antibody and the protein have complementary shapes. So you have to have these things fold up correctly for them to work. That's why mutations in DNA are so damaging, because if you change the DNA, then you can change the amino acids that get put into the polypeptide, and then it'll fold up incorrectly and it won't work. So it's really helpful to be familiar with the 4 levels of structure in proteins. So please make sure you can conceptualize this and you're solid on this. Primary structure is critical. That is simply the order of the amino acids. So the primary structure is basically which amino acid goes first, which is second, third, and so on and so forth. And again, that's dictated by DNA. Now, secondary structure comes into play when you have the skeletal elements of amino acids interact with one another and form hydrogen bonds. So remember, amino acids differ in their R groups, but the skeletal components have oxygen and nitrogen, right? So they are sort of electronegative. So what you can do is get what are called alpha helices or beta pleated sheets. And that can make the polypeptide start to take on a bit of a three-dimensional shape. Now, the rubber meets the road with the tertiary structure. With the tertiary structure, the polypeptide is folded up and has become a functional protein. So tertiary structure is really important because it's all folded up and now it's ready to do things. Tertiary structure happens when all those R groups that are unique interact with one another. So things can happen in all sorts of different ways, right? Hydrogen bonds can occur. We know ionic bonds can happen, right? You can have charged particles on these R groups. And what they can do is either, if they're ionic, they'll be in contact with water, they'll be hydrophilic. And if they're hydrophobic, then they'll fold up to avoid water. Van der Waals forces, again, polypeptides are big, so that can help stabilize the protein. Another interesting feature is what's called disulfide bridges. There certain amino acids have R groups that have a sulfur in them. So that sulfur is why like rotting meat and rotting eggs smell. But the sulfur-sulfur bonds are also pretty stable, so it helps stabilize the protein. So this is a nice image from the book. The purple part, that would be the sort of skeletal part of the amino acid. It's your alpha carbon, amino group, carboxyl group, all that business. And then the stuff that's branched off here, these are representing the R groups or the side chains. So there's your disulfide bridge. You can have ionic bonds form. Here, you can have a hydrogen bond. So different types of interactions can cause that polypeptide to fold up and become a protein. Quaternary structure happens sometimes. Quaternary structure is when you take different polypeptides and link them together into a new, bigger protein. And again, with emergent properties, you make these new, larger proteins that can do different things. A couple of classic examples, collagen. If you look at collagen, it's 3 polypeptides that are interwoven. And since they weave around one another like that, they become really tough. So again, that's the connective tissue that holds our bodies together. If anybody likes beef jerky or turkey jerky or anything like that, if you start to pull it apart, you see some like white fibrous stuff there. That's the collagen that holds it together. Hemoglobin is another good example. Hemoglobin is what's called a globular protein, sort of like a blob, and it's got 4 subunits. So there are 4 polypeptides that link together. So what it does is it allows you to carry 4 oxygen molecules in a single hemoglobin. Fun facts, a single red blood cell contains about 250,000 hemoglobin proteins. Each of them contains 4 oxygens. So a single red blood cell can carry about 1 billion oxygen molecules. So it's a lot. Okay, this chart is going to be really important for you to be able to look at and make sense of. These charts highlight the R groups of proteins, and it allows you to sort of see where they lay or where they go in terms of their behavior, their chemical behavior. So, of course, there are the skeletal parts, the amino group, alpha carbon, carboxyl group, hydrogen buddy, and then the R groups are what are highlighted here. So the non-polar proteins, if you look, it's a bunch of carbon and hydrogen, right? For the most part. Mostly carbon, hydrogen. You do see a sulfur sneak in here and there, and there's this one case where you have a nitrogen. But by and large, if you see carbons and hydrogens, that is a non-polar R group, and if it's non-polar, it'll be hydrophobic. So those R groups would tend to fold inside the protein to avoid water. If you look down here at polar amino acids, you'll see oxygen and nitrogen. So with the oxygen and nitrogen, they're more electronegative again, and then they cause R groups to be polar. So those amino acids tend to be hydrophilic or water-loving, so they'll turn toward the water in a cell. These charged R groups, they tend to either release or take on protons, and as a result of that, they are charged, and they are going to be hydrophilic as well. So having the correct amino acid is really important because those R groups give them like a sort of chemical behavior. They tend to either avoid water or move toward water. And I can think of the relevance of this back, I think it was like 11 years ago, 12 years ago. My wife was diagnosed with breast cancer. And since she was pretty young, she was only like 29 or 30 at the time. Since she was pretty young, they did her genome sequencing on her, or they sequenced just not her whole genome, just that gene, the BRCA1 gene in particular. And when they looked at this BRCA1 gene, they said, okay, you've got a mutation at a methionine, 500 base pairs in. So one amino acid was swapped out for another amino acid. So when we know this, we know there's a mutation. The thing is, so few people have that exact mutation. They said it was of indeterminate significance. They knew there was a mutation there. They just didn't know whether it was harmful or had no impact. So, we're sort of stuck because we just don't know. There aren't enough people with that particular mutation that have breast cancer. So, Chris and I are talking and we're looking through these charts, and, the amino acid that was substituted is in the same nonpolar category. So it sort of gave us some idea of, what sort of treatment options may be best for her, given, the chances of what it would be that it would be a deleterious or harmful mutation. Now, that said, my wife's healthy as can be. She's going to live forever, so she's in good shape. But it was an example of, hey, you know, doctors and researchers only know so much, and you have to try to make decisions on treatment based on the information you have. And if you have some basic understandings of, how biology and biochemistry work, it helps you sort of understand where you're at in the process. All right. So I thought I've found this a number of years ago. I think this is a really fun animation to look at how R groups interact to cause a protein to fold up. So let me see if I can get this working here.
Writing's not that easy, but Grammarly can help. This sentence is grammatically correct.
Here we go. So you can watch the animation here. So the yellow is the pleated sheets, and then the alpha helices are those spirals that you see. This is a collapsed view, but these sort of geometric shapes, these are the R groups interacting. So the ribbons are the skeletal parts of the polypeptide, and then the sort of jagged R groups are the geometric shapes. Those are the R groups. So it looks at how they interact. Trying to become more stable. Cool. See that? How fantastic, right? So do you see the part where the chain sort of folded into the middle? Well, what can you guess about the R groups in that portion of the polypeptide that folded into the middle of the molecule? If it's folding in like that into the center of the molecule, it's trying to be away from water. All that black space that you see, imagine that's the water of the cell, right? So non-polar portions of the polypeptide, I can't believe I said that so easily. Those non-polar portions sort of slide into the middle and seek to avoid water. And then the R groups that you see on the outside of the molecule, you can assume that they're hydrophilic in that they would be in contact with water. So those R groups really matter because they make these polypeptides fold up and take on certain shapes. So I just think that's really cool. I forgot to mention, the sort of bubbly looking things there, those are called space filling models. So here they have some collapsed models that sort of show the different atoms that go into the R group. And then the space filling part is sort of like how much space they'd actually fill up and what would that look like. So a really cool animation. All right. Now, again, going to why primary structure matters. If you change the order of the amino acids, you can actually change how the polypeptide folds up. And if the protein isn't working, well, then that can be really harmful to the person in particular. And a great example of this is sickle cell disease. And in most cases of sickle cell disease, a single amino acid being swapped out causes change to red blood cells and make them sickle or have sort of a crescent shape. And when that happens, they don't deliver oxygen to tissues, they clump together, and it can shorten a person's life by years or even decades. So here are images that show the primary structure. You have it fold up into a three-dimensional protein, and then again, the hemoglobins have a quaternary structure, so there are four of the polypeptides linked together. Well, if you swap out, I think it's like a valine for a glutamine or something like that. If you swap out one amino acid for another, you can see how this sort of folds in. So you swap out something that's hydrophilic for something that's hydrophobic. That changes how this folds up, and then you get this sickling of the amino acid. So, a single amino acid being swapped out can have life-altering effects for a person. Now, next semester when we get into biotech, we'll look at how there have been some people where it looks like successfully they've been able to actually alter their DNA and produce healthy red blood cells. So it's sort of amazing. So you guys did a lab where you looked at the idea of denaturing proteins. So proteins have what's called a native shape, or sort of their functional shape. And what can happen is if you break the bonds between the R groups, you can cause the protein to denature or lose its three-dimensional shape. So if you do that, you can basically destroy the shape and therefore function of the protein. So what can do that? Change in pH, change in temperature. Sometimes ions like salts can bust that stuff up. But just recognize that if you have some sort of environmental stressors, you can basically break the interactions between the R groups of the proteins and destroy its shape. Now, one thing about proteins is, in a lot of cases, we don't know the exact shape of the protein. This is getting figured out more and more rapidly just because of techniques with being able to visualize protein structure. But there is a big gap between understanding the primary structure of amino acid and how all those R groups might interact to create something that's stable. So there's actually a software program called Foldit. I think it was developed at the University of Wisconsin. And what they do is they use gamers and citizen science. So they take these primary structures and you get sort of like an image, somewhat like you saw in the animation that you saw there. But you can click and drag on things and sort of rearrange the polypeptide and try to find something that's more stable. And it's cool because like when you get something that's more stable, you like earn points. And if it's less stable, you get fewer points. And so what they've actually done is written scholarly papers where they've determined the three-dimensional structure of proteins from people playing the video game. People have folded up until it gets something that's like, this seems pretty stable. And then researchers can use the more expensive equipment to try and test that. And they've actually found three-dimensional structure of proteins by playing games. One other thing to mention, chaperonins. These are sort of interesting. Proteins, polypeptides are produced in the cytoplasm of the cell, right? Well, when you get an image of a cell, it's always stationary. But in reality, things are bouncing around like crazy. So there's all sorts of stuff that could interfere with the polypeptide folding up. So polypeptides could really benefit from being like semi-isolated while they're folding up into their native shape or their functional three-dimensional shape. So there's a little chamber, or like a reaction chamber called a chaperonin. And chaperonins allow polypeptides inside of them and keep them from being bombarded from everything else inside the cell until they fold up and get into their sort of native shape and they're ready to go to work. So that's it. Proteins, they're amazing. But what I would encourage you to do, think back over the entire chapter. Chapter 5 is the bulk of the test. I'll be honest with you. Chapter 5 is really where it's at for the test. Think about what atoms you find in these different types of molecules, be able to identify the different types of molecules, sort of know their relevance inside of cells, and that should take you a long way. All right. Trying to think of anything else. Oh, what things are made of? Carbs are CHO with a lot of O, right? Lipids are CHO with very little O. We said nucleic acids were CHOMP because they've got the sugar phosphate backbone and nitrogenous bases. Proteins you can identify because they are CHONs, carbon, hydrogen, oxygen, but they also have nitrogen for their amino groups and then sulfur for those disulfide bridges. If you see sulfur, you're dealing with a protein. All right. Whew, hey, you made it. Congratulations. Thanks for sticking with it. Summarize those big ideas, and you're all set. Thanks, folks.